Validation of the shoot assay as a proxy to determine bud dormancy in intact apple trees. Over the past couple of years, dormancy research has moved from studying the release or exit from dormancy to studying the induction or entrance into dormancy. This situation has arisen due to the realization that problems arising in bud dormancy in trees that are grown in areas of inadequate chill actually already begin in the induction phase. The buds simply do not go dormant properly. To try and rectify something during the release or exit from dormancy, which has actually already occurred in the induction or the entrance into dormancy, is very much like trying to take BioPlus in the morning if you've had a bad night's sleep instead of actually taking a sleeping pill before you go to bed. We have in the past, like all other international research, used an excised defoliated distal shoot section to study dormancy progression or the depth of dormancy. We take these shoots, we put them into a bucket, we force them at 25 degrees Celsius with 24 hours daylight. And the time interval it takes for half of these shoots in the bucket to start budding is used as an indication of how deep in dormancy the buds are. We take that value and we plot it here on the y-axis against the day on which those shoots were collected in the field, and we get a graph. This graph we call a dormancy progression, and we use this to compare different treatments, different cultivars, or whatever we want to compare or to research. However, if we think about it, in the exit of dormancy phase, these shoots look like this in the field anyhow. They do not have leaves and the root system is not working. However, during the induction phase, we are dealing with shoots on the tree which have leaves and there is a root system which is functional. Now we want to know with all honesty, can we be sure that we are dealing with the same thing and we can use the assay in the same way? So how did we go about investigating this question? Well, we used 1,5 meter whips of 793 grown in bags, 5-liter bags from the nursery. We transported them to our growth chambers in Stellenbosch. We then divided them into 4 different treatments. Now, the first treatment was the intact plant. The second treatment, we defoliated the plant. The third treatment, we excised a part off the plant and we used it as the excised shoot, and then we would defoliate that and have it as the excised defoliated shoot section, which is actually our assay. For the experiment, we had 8 different treatment dates from April, when it was bud cessation, until the 27th of August. We had 2 replicates of 8 shoots each, and then we forced them and we produced our dormancy progression curves. So what did we find? We found that irrespective of whether there were leaves or no leaves, the root effect, roots tended to increase the time to 50% bud break during the induction phase and decrease it during the release from dormancy. It happened in both cases. The leaf effect, irrespective of whether there were roots or no roots, when there were leaves on the trees, it would increase the time to 50% bud break up until 100% leaf drop. We can also see that closer to 100% leaf drop, the effect becomes less there and there. These little red dots that I've included here, this is a phenomenon in excised shoots in buckets that occurs under certain conditions. I'm not going to speak to you about it at the moment, but it does mean that it actually increases the time. So these should most probably have been about like that. So what can we conclude at this stage of this project? We can conclude that the root system appears to increase the time to 50% bud break during the entrance phase and decrease the time during the release from dormancy. Leaves appear to increase the time to 50% bud break during the entrance phase. The leaf effect is less, closer to leaf drop. At this point in time, we've got to start looking for correlations between these effects. but we need more years data before we can do this. Thank you very much for listening to my talk.